growth. Kasi meron din eukaryotic growth at saka prokaryotic growth. Kasi when we talk of eukaryotic, we're talking about the cell size. Lalaki lang siya. When we talk of prokaryotic growth, that's more into increasing in cell number. So that we define growth as the orderly increase of all chemical constituents of the cell. So it is a process that entails, sabi ko nga kanina, increase in number. So it is a process that entails replication of all cellular structures, organelles, and protoplasmic components from the nutrients present in the surrounding environment. So, kailangan, lahat din ng kailangan ni bacteria, ni organism, nasa environment niya para siya ay able to grow. Okay? So, we have here now that the requirements for microbial growth can be divided into two main categories. So, meron tayo ng physical at saka nung chemical requirement. Now, physical, this would be already your temperature, yung pH, and osmotic pressure, whereas chemical will be on the sources of carbon, nitrogen, sulfur, phosphorus, oxygen, trace elements, and organic growth factors. Okay, so I will start first with the chemical requirements. So, ano yung chemical requirements niya? So, when we talk of the chemical requirements here, we're talking of those that like the carbon, nitrogen, and also the source of energy. So, ito yung major nutritional needs ng bacteria for growth. Okay, same with us. So, pag sinabi natin source of carbon, now, ang carbon, this is used for making cellular components of the bacterium. So, syempre, pag hindi mo mabuo lahat yung cellular components niya, hindi naman siya mabubuhay. And this makes up 50% of the dry weight of a bacterium. Whereas nitrogen, this is used for making proteins, and it makes up 14% of the dry weight. And of course, the source of energy, which is also needed in performing cellular functions, and that would also be your adenosine triphosphate. So we'll get into that one by one. So aside from what has been mentioned earlier, sabi ko nga, source of carbon, source of nitrogen, and then you also have small amounts of molecules like your phosphate and sulfur. Both of these which makes up an additional 4% of the weight, and the phosphate is for the nucleic acids and phospholipids of the cell membrane, whereas the sulfur is for protein synthesis. Now, there are also various metals and ions, like for example, you have here mineral ions like sodium, potassium, chloride, and calcium. And wala siya. Okay, so let's start with the carbon source. When we say carbon source, now you are now again trying to classify the bacteria, either they are heterotrophs or are they autotrophs or lithotrophs. So ano naman yung heterotrophs? If you remember, dito kasi sa mga grouping, madami kayong ma-encounter na terminologies. So, yung mga prefix pa lang, alam nyo na kung ano yung hetero or your organotrophs. Ito yung mga nagre-require ng organic carbon for growth. So, ayun yung organic carbon for growth, because trophs is like to feed, so that they will need now, ang source ng carbon nila would be from yung organic carbon. And they can also use reduced preformed organic molecules from other bacteria. When we're talking kasi of organic carbon or itong preformed or organic molecules, pwede dito yung glucose. Okay? Nandito na rin siya sa chemoheterotrophs. So glucose can both serve as a source of carbon and also as a source of energy. Yung mga chemo, again, you have there the prefix chemo. Yung chemo is always from chemicals. Heterotrophs, so they get most of their carbon from the source of their energy. What are those? You have your proteins, carbohydrates, and lipids. So that yung pag-derive nila ng energy from chemicals, you will need to consume other organisms in order to live. Like for example, dito sa ating heterotrophs. Ang example natin dito could be your E. coli and Pseudomonas aeruginosa. Kasi ito, they can use wide variety of organic compounds. This is also the reason why they are very easy to grow in the laboratory. Kasi simpleng media lang ang kailangan nila. Simpleng nutrients lang ang kailangan nila, tutubo na sila. Okay? Now, for our autotrophs, sabi na naman natin, o sabi ko, you always look into the prefixes of all of these terminologies. Pag sinabi nating auto, di ba? Pag sinabi nating auto, ano yung pag sinabi nating auto? What does the prefix auto always means? Anyone? Hello? Ano ibig sabihin ng auto? Nakita nyo siya sa autolysis. Now you see it as autotrophs when we're talking.
Self.
Yes. Self. Yes, self. Like, they can make their own. Diba? Hindi nila kailangan. So, kaya nilang gawin yung sarili na lang kailangan na biological materials at saka yung energy na kailangan nilang for growth. So that you also have now your chemo-autotrophs and then you have your photo-autotrophs. Sabi nga natin, auto, sarili ko. Kailangan gagawin ko yan ng sarili ko. But now, when you put the prefix chemo and the photo doon sa inyong autotrophs, saan manggagaling yung pangangailangan nila na yun? Saan nila i-derive yung carbon na kailangan nila? So, they get it from pag chemo, sabi natin kanina, chemicals. So, pag photo, saan manggagaling naman yung energy nila? From photo.
Light source.
From light. Very good. Like for plants, it is photosynthesis. So that they have the process of producing their own food. So that when we say also, in other books kasi, sabi niya sa mga ibang journals, they always have to interchange chemolithotrophs from chemoautotrophs. So that, kita mo naman sa taas, we're saying that its autotrophs is also the same with lithotrophs. Okay? So that when we say here, chemolithotrophs again, palitan mo lang naman yan din. Kasi dito, use an inorganic substrate such as hydrogen or thiosulfate as a reductant and carbon dioxide now as a carbon source. But if you get into some books, they said that yung earlier journals, ang ginagamit nila, instead of chemoautotrophs, they were using chemolithotrophs. So, that's with the carbon source. How about for our nitrogen source? So, anong si nitrogen? You said that nitrogen is a major component of proteins and nucleic acid of a typical bacterial cell. So that the end product of all pathways for nitrogen assimilation is the ammonium ion. And most microorganisms use this ion, ammonium, as a sole nitrogen source, and many organisms possess the ability to produce it from amines. So nakita nyo dito, you have different processes. You have your nitrogen fixation, you have ammonification, and then you also have your denitrification. So when we say nitrogen fixation, it is the ability to assimilate nitrogen reductively via, pag NH3? Anong NH3? Hello?
Ammonia.
Ammonia, okay? So that ammonification naman would be the production of ammonia from the deamination of amino acids. Then, of course, when you say denitrification, you have again, dito nyo ma-apply lahat yung medical terminologies nyo. When you say denitrification, it's the conversion of ammonia to N2 to nitrogen under anaerobic conditions. So, phosphorus source, di ba na-mention natin kanina, you also have the phosphate and sulfur. So that phosphate as a component of your energy, source of energy, ATP, which is for nucleic acids and coenzymes like your NAD and NADP and flavins. And your sulfur source, autotrophic bacteria can oxidize it to sulfate. So that now here, you take note of this one that some microorganisms can assimilate hydrogen sulfide directly from the growth medium. These growth medium are those culture media that we will be using in the laboratory to let our microorganisms grow. So that example of this one, which you will learn later on in the different media that will be used in the laboratory in the culturing. When you use culturing kasi pag nasa microbiology tayo ng bacteria, would be like your SIM medium. This is your sulfide indole motility medium. So makikita mo doon ngayon ang production ng H2S. Okay? So, for minerals and mineral sources, di ba sabi natin kanina, aside from your sulfur and phosphates, there are also these metal and ions that also play a role in the enzymatic activity of the organism. So that what are your mineral sources? You have your magnesium ion and the ferrous ion. This is for enzyme function. And of course, you also have your magnesium and potassium, which function and for the function and integrity of ribosomes, and then your calcium, which is a constituent of gram-positive cell walls. So you also have other minerals there that we need to provide for the microorganism depending na sa kanyang needs. You have there your Mn, manganese. Tama ba ako? Mo is molybdenum. Ano ang CO2?
Carbon dioxide.
Hindi hindi. CO maliit.
Cobalt.
Cobalt.
Global.
Cobalt?
CO maliit. CO maliit. Ayaw kasi pag sa PowerPoint dito, hindi ka maka-superscript. Hindi siya aakyat yung mga 2 niya or bababa, parang ganun. So that Cu is copper, di ba?
Yes, ma'am.
And Zn is zinc.
Zinc.
Yes, ma'am.
Tama? Okay. Lahat ito, di ba nakikita nyo sa Centrum pag sinabi nila multivitamins natin? Napatawa ka tuloy. Okay. Next one, growth factors. What are the other growth factors that we need? Pag sinabi natin growth factors, hindi ito yung parang sa atin. Ano na yun? Chlorella ba yun? Yung nasa cherry fur? Ano yung nasa cherry fur? Diba yun yung mga growth factors natin para tayo daw ay tumangkad. Di pa uso yun nun, kaya hindi ako tumangkad. So what are these growth factors? Napatawa ko, baka tulugan nyo ako. Ako'y napuyat ng pag-check bakit hindi lumalabas ang mga answer nyo kagabi. Okay, these are organic compounds that are necessarily needed by a bacterium in order to grow. Now, these substances are usually provided in the culture media. An example of this that can be present in your culture media, you have amino acids, pwede rin purines, pwede rin pyrimidines, and vitamins, hemoglobin. Saan manggagaling ang hemoglobin natin usually? If we're putting it into our media, like for example, ano example nyo dito? Yung ginamit nyo sa inyong preparation. Zarina. Anong culture media yan? Yung may hemoglobin. Anong culture media? Any idea? Or saan makukuha si hemoglobin? Anyone? Anyone? Any two? Asan na kayo? Okay. Si Domingo lang nandyan sa ano? Nasa group? Nasaan yung iba? Kaso binawi niya, charmam. Sabi niya, yes, blood agar. Because then, from the blood, you'll get the heme that will now provide the hemoglobin for the organism. Okay, so that under your growth factors, again, now you will have to classify them into either their phototropics or oxotropics. Correct ko nga, baka nagkamali ako sa ano, sa encode ko. Okay? So when we say prototropics, what are these? These are those who do not require exogenous sources of growth factors since they synthesize their own. Whereas yung mga oxotropics ninyo, ito yung mga nagre-require ng addition ng growth factor to culture media for, kung makita yung ilalim niya. Sorry, for growth. Okay? Anyway, for the note here that all bacteria that inhabit the human body fall into the heterotrophic or organotropic group. Why is it? We will try to explain now why that all bacteria that inhabit the human body fall into the heterotrophic or the organotropic group. Anyone? Except Domingo or except Dexter. You can unmute for a while just to make your answer. Ano ba yung ano natin ng heterotrophic or organotropic group? Hi. Hello. Who can explain this? That all bacteria that inhabit the human body fall into the heterotrophic or organotropic group. Why is that so?
Ma'am?
Yeah, go ahead. Game. Yeah. And so, ganun. And you just try to. So if what are heterotrophs? You start right now trying to say what are sabi mo nga they require an organic form so that when they inhabit when they get into the human body, so saan nila kukunin yun? Ipro-provide mo ngayon doon sa bacteria yung kanyang ano ipro-provide mo sa kanya? Ano, teka, sino yun? Si ano ba yun? Si Zarina? Sino yung nagsagot? Hindi ko makita.
Si Zarina, ma'am.
Oo, Zarina. Game. Ano yung heterotrophic muna? Ang ballpen ko. Ang ating recitation. Ja. Heterotrophic. Kevin. From other organisms so that when we say that lahat ng nag-i-inhabit sa atin na mga ano, they're heterotrophic. So that it could, when you conclude there that the carbon source is provided for din sa product ng metabolism natin. Okay. Next, fastigious bacteria require additional substances such as vitamins, purines, pyrimidines, and hemoglobin for growth. What is again fastigious bacteria? Except yung mga una na. Except Dexter, except Kevin, except Zarina. Except. What are fastigious bacteria? So that we just get to review. Umaya, yung mga note na yan ang lumalabas sa mga exam ninyo. What are fastigious bacteria? Anyone? Anyone?
Ma'am? Ma'am,
Yeah.
lang.
Sino to?
Ma'am, Maridine Pagatpatan po.
Ha?
Maridine Pagatpatan po, ma'am.
Maridine. O sige.
Ma'am, ang fastigious bacteria, sila yung mga bacteria na may special requirements na kailangan bago sila mag-grow po.
Okay, so fastidious na mention natin yan doon sa ating bacterial structure, bacterial, doon sa ating previous lecture, that ito yung mga nangangailangan ng additional substances na. Kaya nga nandyan, you require additional substances such as vitamins. Okay, so no need to ask you what are saprophytes, alam nyo na yan. Okay, so how about with the oxygen requirement? When we say of oxygen requirement, ano naman si oxygen requirement? So that we say now here, you classify them again as either sila ay aerobe or sila ay anaerobe. Okay? So that under that din, aside from them being classified as aerobe or anaerobe, meron din tayo ngayon again your obligate and your facultative and your facultative anaerobes. Now, looking at the definitions there, now can you try ala na, nag-cramps yung pa ako. Can you try to differentiate one from the other? Parang ano lang, in your own words. Sino susunod? Except yung mga nauna na, nalista ko na kayo. Yes, sino to? Sorry, hindi ko makita kung sino yung nag-uon ng ano.
Tamparalag, ma'am. Tamparalag. 20.
Atam.
Ma'am, from the word itself, obligate parasites, ay hindi. Obligate
Wala pa
aerobes.
sa parasite ngayon, nasa microbiology na
Hindi,
tayo.
ma'am, nadulas lang. Okay, so
Okay.
from the word itself, ma'am, obligate, tapos aerobes, ibig sabihin, they require oxygen. Ibig sabihin, hindi sila mabubuhay kung walang oxygen sa kanila. Ngayon
Mhm.
naman, yung sa obligate anaerobes,
Hindi.
they are,
Tam,
they are-
hindi. Tam.
oh, ma'am.
Tam. Sundan mo yung nasa slide natin. So, okay ka na sa obligate aerobes. Ngayon, after aerobes, you also have your facultative anaerobe. Game.
Okay, ma'am.
Game. Tuloy mo yung ano mo. Tama ka na. Next. Tuloy mo yung ano mo. Yung,
Ma'am, yung facultative aerobes naman, ma'am.
Darling ko. Basta tignan mo yung aking slide, ha? Okay lang yan. Kasi ganyan din ako kanina. Sabi ko, so you have said that obligate aerobes, ito yung mga organism na tutubo lang kung may oxygen. Ang next dyan is you have your facultative anaerobe. Ano yung facultative anaerobe natin?
Ma'am, ano yung gagawin ko, ma'am?
I-explain mo lang siya. Ano si facultative anaerobe? Hindi, kasi teka lang. Sorry. Kasi na ano ka kunya, alam ko yan dapat obligate aerobe and then facultative aerobe. Di ba? Pero dito kasi sa slide, you are talking about the growth in the presence of atmospheric oxygen with aerobes. Tama yun? Baka na wala kayo sa akin. Baka mawala kayo. Now, we're talking now of obligate aerobes. Sabi mo nga, ito yung mga mag-grow lang pag merong oxygen.
Oxygen.
Okay. So, after that, dito kasi nilagay ko naman si facultative anaerobe. So, ano naman si facultative anaerobe? By the word facultative muna. Kasi, di ba, maalala
Ma'am, facultative anaerobes, ma'am. They can grow in the absence of atmospheric oxygen. Ibig sabihin, facultative. Kaya nila na walang oxygen sa buhay nila, ma'am. Sa growth nila.
Sige. Kaya nila, kasi di ba sabi natin noon, if you say is obligate, kailangan nandyan yan. Yung kailangan ko. Obligate aerobe, kailangan, kung sabi natin aerobe is the growth in the presence of atmospheric oxygen, so that kung sinabi kong ako ay obligate aerobe, I need then, it is a prerequisite for me that atmospheric oxygen should be present in order for me to grow. But when I say I am a facultative anaerobe, these are fundamentally, take note of the word ha, these are fundamentally aerobes but can grow also in the, but can grow also in the absence of oxygen. That is why nakalagay dito sa aking slide is that it can use oxygen when it is present but are able to continue growth by using fermentation or anaerobic respiration when oxygen is not available. So, kasi dito, pag intindi natin, wala naman siyang oxygen pero tumubo din siya. Pero still, kasi din facultative, di ba? Kung tignan mo yung facultative and wala dapat. So that it can grow when oxygen is present, can use oxygen when it is present, but are able to grow by using fermentation. So, iba din na source ng oxygen. Okay? So, para hindi kayo maguluhan mamaya, tignan nyo lang yung kasi pag nagpa-exam ako, itong anaerobe na ito, maano nang ma-mix nyo with your facultative aerobe din mamaya. So that now we get into your microaerophils. Again, you have there your prefix micro. Dapat mas maliit siya. So that it require a reduced level of oxygen to grow, which is about 2 to 10% lang of oxygen. And this environment may be generated in culture jars or pouches using a commercially available microaerophilic atmosphere generating system. So that the percentage here, when we say meron siya kasing kasama na carbon dioxide. So 5% of oxygen, 10% carbon dioxide. Then you have there your 85% nitrogen. Okay. So you have there your examples again. Now, doon yung classification natin sa aerobe. So sa aerobe, you have your obligate aerobe. You had your facultative anaerobe, which are those that they are fundamentally aerobe pa din. Okay? But they can grow in the absence of oxygen. So those are your facultative anaerobe. And then you have your microaerophils. Now, how about your anaerobes? So again, you have there your prefix, which means that wala siyang oxygen. So these are the microorganisms now that they grow in the absence of atmospheric oxygen. So sabi ko sa iyo kanina, tignan mo na naman dito. So you have your obligate anaerobes, then you have your aerotolerant anaerobes, or this could also be again, tanggalin ko na lang siguro yan para hindi kayo malito-lito later on, or your facultative aerobes, because when we say facultative aerobe, these are also your fundamentally anaerobe organisms. So we'll stick to, para walang confusion, we'll stick to facultative anaerobes under your aerobes, and for anaerobes, we'll use your aerotolerant anaerobes. This is only good to know because maybe some books will be calling them facultative aerobes and some books will be calling them aerotolerant anaerobes. So for purpose of standardization, parang ganun, basta when we will have our exams or any quiz or whatever, I'll be using your aerotolerant anaerobes, and for the anaerobic bacteria natin, you'll be using your facultative anaerobes. Okay? So obligate anaerobe na naman, sabi natin, they are unable to use molecular oxygen for yielding or energy yielding reactions. So they lack both your superoxide dismutase and catalase. What is this your superoxide dismutase and catalase? I will explain that to you later. Andyan yan sa growth ng bacteria dun sa mga tubes. But nevertheless, this catalase is your enzyme, as also your superoxide dismutase. Or kung meron na nakakaalam mamaya, pa-explain ko ulit sa inyo. Whereas your aerotolerant anaerobes, they can grow in its presence, but they do not use it as a hydrogen acceptor. So that dito din yun sa fermentation ng carbohydrates, this can be now a source. Instead of the oxygen, then they can do fermentation also. Okay? This will be sa Enterobacteriaceae later on. So anaerobes use now, how do you maintain this environment for your microorganism to really grow so that you can use reducing agents like your thioglycolate, and the tubes of agar can be sealed with a layer of petrolatum and paraffin. This petrolatum and paraffin can now avoid the entry of oxygen into your media. Okay? Makita nyo mamaya din yan sa ating tubes. And then the culture vessel can be placed in a container from which the oxygen is removed by evacuation or by a chemical. So I'll try to take out the oxygen. And this is the most common that is used usually for anaerobic cultivation, is that organisms can be handled within an anaerobic glow box. Okay? So that obligate aerobes, and take note of this one because you need to know ano ba ang role ni superoxide dismutase at saka ni catalase. Kasi these enzymes now try to get rid of the oxide, which when it not eliminated will be toxic to the cell. Kaya meron tayong tinatawag na oxygen toxicity. Okay. So, ito yung sinasabi ko sa inyo. O sinong pwede mag-explain yan ngayon? Just by looking, kasi mamaya meron kasunod na slide yan. Okay. Just by looking now, as you see here, this is the first tube is, yung lang babasahin ko. The first tube is for your obligate aerobe. Ano na ulit ang obligate aerobe? Anyone? Recall, obligate aerobe.
Needs oxygen po, ma'am.
Sino to?
Brandon Arzadan.
Brandon. Okay. Brandon, since you said that your obligate aerobe are those that need oxygen to grow, explain mo itong first tube.
Ma'am, kaya po siguro nasa taas sila, ma'am, kasi mas malapit sila sa air, ma'am, compared pag nasa pinakailalim sila, ma'am.
Okay. So that, dito, sige, mamaya na natin i-generalize. Next, facultative anaerobe. Except yung mga nag-recite na kasi nilagay. Next, sino to?
Nolan, ma'am. Nolan Manawis po.
Okay, Nolan.
Yes, ma'am. So katulad po ng obligate aerobe, ma'am, itong facultative anaerobe po, they also require they can also require oxygen po. They require oxygen pero they can survive without it. So katulad din po ng obligate aerobe, kailangan din po nila ng mechanism to break down yung toxicity ng oxygen. Ang primary na ginagawa po kasi ng superoxide dismutase is bine-breakdown niya po yung molecules ng superoxide para maging oxygen na lang po. And then yung catalase, ma'am, ang bine-breakdown niya po is yung hydrogen peroxide natin para mapaghiwala yung hydrogen and oxygen. At yung oxygen, may-utilize niya na yung facultative anaerobe natin, ma'am, i-utilize niya na po yung O2, yung oxygen.
Yung
kaya
O2.
Kaya kailangan niya po
Bakit kailangan niyang i-break down di ba, your oxide plus hydrogen will be forming your hydrogen peroxide, di ba? H2O2. So, ano mangyayari pag wala kang superoxide dismutase muna?
Ma'am, base po sa nabasa ko is may chance po na mamatay yung yung facultative anaerobe natin kasi masyado pong toxic. Kasi ang superoxide is toxic, ma'am.
Oo, meron. Yung superoxide is toxic. Pagkakaroon ng oxygen toxicity. Tama? Okay.
Yes,
so that after you form yours, di ba yan, nakikita nyo doon sa first tube, yung oxide, superoxide mo plus hydrogen, now you will form your O2 plus your hydrogen peroxide. But this hydrogen peroxide has to be broken down into water then and oxygen through the action of your enzyme catalase. Then we'll learn about more of this later on sa inyong gram-positive oxide. So, Nolan. Okay, next. Obligate anaerobe. Thank you, Nolan. Obligate anaerobe. Tube 3. Another one, except those who have recited. We have Dexter, Kevin, Zarina, Maridane, Tam, Brandon, and Nolan. Or the rest, anyone. Obligate anaerobe.
Good afternoon, ma'am.
Good afternoon. Sino to?
Ma'am Ira Tumakay po.
Si Alin? Ulit.
Ira Tumakay po, ma'am.
Okay, Ira, go.
Ma'am, yung obligate and aerob po, hindi po sila gumagamit ng oxygen kasi po they lack the enzyme superoxide dismutase and catalase. So, based po dun sa tube 3, reasonable lang po na nasa baba po banda yung obligate and aerob na organisms kasi hindi naman po nila kailangan ng oxygen.
Hindi nila ayaw nila ng oxygen so that they grow dito sa bottom part ng tube because that is where your environment is devoid of oxygen.
Yes.
Okay, very good, Ira. Microaerofield for tube.
Hello, ma'am.
Yeah, good afternoon.
Good afternoon po.
Sino to?
Ria po. Burigsay po.
Okay, Burigsay, Ria. Okay, Ria, go.
So, yung microaerophile po, kaya po parang nasa gitna-gitna po yung mga bacteria kasi nagre-require pa rin po sila ng konting oxygen for growth po.
Okay, so they can live in either parang middle group siya. Kaya makita mo dyan, they still have small amounts of catalase and superoxide dismutase. Okay. And the last but not the least, aerotolerant. Ano lang ang meron siya? Ano yung aerotolerance? Next. Next, sino pa nandyan? Marami pa kayo dyan. Kung tawag na ako. Charlene. Bulan. Where's Charlene? Charlene? Is Charlene here? Asan si Charlene? Yes, Charlene. Aerotolerant. Nakatulog ka na yata eh.
Sila yung ano po ma'am, nag-freeze yung screen ko. Sorry po.
Sige.
Yung mga anaerobic, I mean
Aerotolerant.
yung aerotolerant, they are only anaerobic growth po, but continuous in presence of oxygen. Tapos yung explanation of growth patterns po niya is growth occurs even
Echinella.
Ay, kanela. Ano yung K?
Kinchella.
So, Kinshella. So, considering that they are capnophilic, so what type of bacteria are they? Meron kang enriched with, na word. Pag nakita mo si enriched with something, whatever, vitamins, minerals, and now enriched with extra carbon dioxide, what type of bacteria are they? Ito yung tinatawag natin na ano na? Yung sinasabi ko lagi na ang arte-arte na bacteria, ang dami niyang need. Ito yung ating fastidious. Because your HASEC are your gram negative fastidious organisms. Okay? So that you see here that diagnostic microbiology laboratories often maintain their aerobic incubators at 5 to 10% carbon dioxide level. So most aerobic and facultative aerobic bacteria need only, what you see here is 0.03% of carbon dioxide. So if you see here, sabi natin, how do you provide the 3% carbon dioxide? Is itong candle jars. Usually, we just improvise this one, but we'll also have that sa laboratory. Yung candle jar kasi meron tayong gas pack, meron din tayong ginagawa na improvise lang na candle jar kasi mahal yung gas pack. So, pag yan nilagyan mo ang kailangan ng iyong microorganism, ang carbon dioxide in order to grow, ang ginagawa namin yan sa laboratory, meron kayo ng ano doon, kita nyo yung nag-experiment kayo sa ano na ito? Sa chem ninyo, yung may glass na may cover, parang para sa pagtanggal yata yun ng moisture. Anong tawag nyo doon sa chem? Nagamit nyo ba yun? O wala din kayong experiment ng chemistry? Na bagyo-bagyo naman kayo ng first year kayo. First year ba yun na bagyo-bagyo tayo o second year?
First year, ma'am.
Oo. Naka-experiment din ba kayo sa chemistry? Yung ginamit nyo, parang humidifier yata. Yun yung malaking glass jar na meron siyang parang butas-butas na nakalagay sa loob niya. And then may takip. Nagamit nyo yun? Hindi. Walang nakagamit sa inyo? Hello? Wala siguro, walang sumasagot eh. Wasted candle jar is, pwede tayo mag-improvise yan. So, pag na-strict na yung, inoculate na yung mga microorganisms sa kanyang petri dish with the enriched media, and then you put them into your, alam niyo yung ice cream container pa na na mga lata. Kasi ngayon, karamihan ng ice cream containers natin, plastic na. So, meron yung lata na lagayan, di ba? So, once you put it there, inside, and then lalagyan nyo yan ng kandila, ilalight nyo sa kandila, and then sasarhan nyo sa kandila. Sasarhan nyo yung can. What will happen? What environment are you going to produce? Anyone? Naglagay ako sa can ng kandila. Nilight ko si kandila. Nung nalight ko si kandila, clinose ko si can. What will happen? Mamamatay yung kandila. Pag namatay yung kandila, ano ang nawala at ano ang na-produce? Mababawasan ka ng oxygen, mapapalitan ng
Anaerobic.
Carbon dioxide po.
Magproproduce ka ng carbon dioxide. Okay? Hindi naman siya mawawala talaga ang oxygen mo, pero madideplete ng konti ang oxygen mo, kaya mamamatay si kandila mo. Then you will be producing carbon dioxide. That is our improvised candle jar. Kasi if you're really going to use the gas pack one, na talagang commercially available, medyo may kamahalan kasi yun. Okay? So, we're done with the chemical composition requirements of your bacteria. In summary, who will summarize it? Who would like to summarize from the three major requirements of your bacteria? Sino? Wala nag-volunteer. Alfred, are you there?
Good afternoon, ma'am.
Good afternoon, Alfred. In summary, as to the chemical requirements of your bacteria and able to grow.
Ma'am, hindi ko masyadong marinig yun.
Ha? Ano yung
Ma'am,
i-summarize mo lang, ano yung kanilang chemical requirements, yung tatlo na diniscuss natin.
chemical requirement.
Yung tatlo. Kahit isa lang.
Ma'am, chemical requirements, ma'am?
Yung kailangan nila. As to their source of carbon, anong classification mo?
Ma'am, carbon, ma'am.
Ano yung mga classification pag source of carbon? Alfred.
Ma'am, pakiulit yung tanong, ma'am. Hindi ka narinig.
Sabi ko, di ba, you have our bacteria needs they have chemical requirements in order to grow, and one of them would be the carbon
ma'am.
source. Now, according to carbon source, how are these microorganisms classified basing on their need for carbon? They're classified as what? Dalawang classification lang.
Ma'am, may ano, organotrophs sa ano. Ayun sa? Lithotrophs.
Hmm? Basic.
Chemoorganotrophs ma'am, tsaka chemolithotrophs.
Chemoorganotrophs at saka your heterotrophs or your organotrophs. Okay?
Heterotrophs.
Okay. So, how about the others? That's for carbon source. Ano pa yung iba? Alexandria. As to oxygen. Asan si Alexandria?
Good afternoon, ma'am.
Yes, Alexandria. As to oxygen requirement.
Ma'am, under po ng oxygen requirements, we have aerobes po. Tapos under ng aerobes, we have obligate aerobes, facultative anaerobes, and microaerophiles.
Yeah.
And the next basic classification po is anaerobes. Tapos under ng anaerobes, we have obligate anaerobes and aerotolerant anaerobes.
Okay, very good. Okay, so the last one, when we're talking of carbon dioxide, Estela, May Pagaw. When we're talking of carbon dioxide.
Hi, ma'am.
Yeah, Estela, good afternoon. So,
Ma'am,
carbon dioxide naman.
Ma'am, pag under carbon dioxide, we have capnophilic.
Okay, you have your capnophiles. Pag sinabi natin sila ang capnophiles, which organisms do require this one? Example of your capnophiles.
Ma'am, we have the Hasek and Neisseria gonorrhoeae.
Okay, so you have your Hasek and your Neisseria gonorrhea. Okay, so when you will be asked to just distinguish one from the other, go back to your notes later on about yung sa growth patterns nila in your tube media. Okay? So that the closer you get up dun sa culture media, kasi pag nasa tube, more aerobic ang conditions doon. Pag the deeper you get into your culture media, then the more anaerobic is the environment. So we get into now the physical requirements naman ng ating organisms. So for this, now you have your temperature, then we have pH, and ano yung isa natin? Kanina, nabanggit na. Temperature, your pH, and osmotic pressure. Okay. So that in temperature, later on in the slides, meron tayong tinatawag ng mga optimal temperature and others. So, primary groups on the basis of their preferred range of temperature. So that you have here your psychrophiles or your cryophiles. So these are ang commonly used kasi dito rather than the psychro is your cryo. Kasi nga from the word cryo pa lang yan yung sa malamig. So that these are your cold-loving microbes. And they grow well at 0 degrees centigrade to a maximum of 20 degrees. But if you get into some wag nyo i-confuse yung sarili nyo kasi may minimum temperature, may optimal temperature, may maximum temperature. So that when we say they grow well, that could be their optimal temperature growth. So that with your psychrophiles or your cryophiles, ito yung mga mag-grow well lang when it is 0 to 20 degrees. And examples for this one, you have your Listeria monocytogenes and Yersinia enterolytica. Naalala ko si Sir Cinia. Hindi ko alam kung naabutan nyo na ito. May issue dyan. May gumamit na ganyang name sa social media. Then you have your psychotropes. Okay. Ano naman yung mga psychotropes ninyo? So these are those that the temperature grow well at a temperature between 20 degrees and 30 degrees but grow well at lower temperatures. Okay. Wait lang. Yung aking ano. Okay. This is an important cause of food spoilage. Bakit sinasabi natin dito na they're important cause of food spoilage? Ano ang temperature sa ref? At ano ang temperature na 20 degrees to 30 degrees centigrade? Goodwin. Good afternoon. Goodwin. Goodwin. Wala si Goodwin. Goodwin? Okay.
Ma'am, dalawa pong Godwin D.
Ay, dalawa ba? Umayang pala. Dalawa
Yes,
nga. Tabalbag yung isa? Ano yung isang Goodwin? Umayam. Sino si ano? Ang daming dalawa. Dalawang Princess, dalawang Goodwin. O Goodwin, Umayam.
Yes, ma'am.
Ano ba yun? Kasi sabi natin dito, psychotropes is the temperature optimum between 20 degrees to 30 degrees but grow well at lower temperatures. Now, these are important cause of food spoilage. Bakit sinasabi natin na itong mga psychotropes are important cause of food spoilage? And what is this temperature na 20 degrees to 30 degrees centigrade?
Ma'am, kasi ang food spoilage is parang nagkukos siya sa parang higher temperature. So, ang psychotropes kasi is nag-grow siya between 20 degrees Celsius to 30 degrees Celsius. So, nagkukos siya ng food spoilage since medyo mataas yung temperature, ma'am.
Saan? Saan siya magkukos ng food spoilage pag mataas ang temperature?
Yes.
Yes,
ma'am. Kasi dapat lower ata yung temperature para ma-prevent yung food spoilage, ma'am. So, sa refrigerator temperature kasi, ma'am, is parang negative 18 to 4 degrees Celsius dapat. Yun po yung required na refrigerator temperature. Yung sa psychotropes, ma'am, is nag-grow siya between 20 degrees Celsius to 30 degrees Celsius. So, may possibility na mag-cause talaga siya ng food spoilage.
Asan ka?
Ma'am? Ma'am?
Chi.
Hello po.
Sige, Chi. Chi, ano ang room temp natin?
Room temp, ma'am? I
Pag
think
sinabi,
nasa 27. Ewan ko, 27 to 30 yata, ma'am.
okay,
Ata, ma'am.
27 to 30. Hindi, mga 30 ka muna lang.
Apo.
So ano ang ano natin dito? That's why, pag sinabi natin cyclotrophs, we're talking of the requirement for growth. Kaya
Yes,
sabi
ma'am.
ano dito, important cause of food spoilage is because they grow well at that temperature. So that, pag nasa labas lang sa pagkain natin, tapos babalik tayo sa spontaneous generation o kaya sa germ theory of disease, balik kayo ng history ninyo. So that, when you put your food outside under room temperature, that's an optimal temperature for the growth of these cyclotrophs. Kaya nga nagkakaroon ng food spoilage because of the growth of these cyclotrophs. Okay? So that we have also our mesophiles. What are our mesophiles? Okay. Yung tinawag ko kanina na crinose ko ulit. Tanggalin ko. Jamina.
Hello, ma'am. Hello,
Okay.
ma'am.
What are our mesophiles?
Mesophiles po, sila po yung mga bacteria po na nag-grow best at the optimal temperature of 20 to 40 po yung nandito sa 20 to 40 po.
Mm-mm. So that this, if they grow well in 20 to 40 or an average of 30 to 37, kaninong temperature yan?
Yung temperature po ng environment.
Kaya nga, sinong environment kaya ang pwedeng merong temperature na ganyan?
Mga may init na lugar po.
Hindi 37,
Mga
di nga mainit yan. Di ba mainit ba yan? Nag-50 nga tayo sa
24.
Tugigaraw eh. Kaninong temperature yan? 30 to 37. Pag pumapasok ka ngayon sa mga department store, chine-check ang temperature mo. Ano ang usually ang temperature mo para papasukin ka?
37.
Hindi, ayaw na nga nila ng 37. Dapat below 37, 37 pataas. Ano eh. So this is our temperature. So that you will discover later on. Di ba ang temperature natin yung 35, 36, wala kang lagnat yan. Yan yung regular daw. Normal temperature natin because if you get up to 37 up, yun na yung may lagnat tayo, hindi na tayo papapasukin kasi baka may dala-dala tayong COVID na dala-dala natin si SARS-CoV-2. Okay? So that you will learn later on that yung mga nag-i-infect sa ating mga ng human beings are the mesophilic bacteria because they love our temperature and they will grow best in that temperature. Okay? So you have now your thermophiles. These are your heat-loving microbes. And you take note of this one. Why is your Bacillus stearothermophilus the example for this? And this Bacillus stearothermophilus is the biologic indicator that is used to test for the effectivity of our autoclave. Alam niyo kung ano yung ating autoclave? Are you familiar with the autoclave? Anyone? Dave Ikin? Ay, meron pa ako na-cross pala dito. Si Chisel at saka si Chunafi. Dave muna, si Dave. Asan si Dave? Dave Ikin. Where are you? Ano ang ano? Alam mo kung ano yung autoclave? At bakit? Nakita niyo yung autoclave sa ating ano? Sa ating laboratory?
Yes, ma'am.
Nagamit niyo ba yun nung last year
Hindi po.
Hindi niya pa nagamit. Okay. Are you aware of the principle ng autoclave?
Hindi po gano'n. Pero ang sa pagkakalam ko lang ang ano po, kinikil niya po yung mga harmful bacteria, viruses, fungi, yung mga ganun lang po.
Okay. So that it is used for sterilization. Okay. So, kinikil niya lahat when you're talking of, when we get into this infection and sterilization anyway, so that your autoclave will eliminate all types of living forms. Okay? Except for some, pero ano na yun, we'll discuss that in our disinfection and sterilization. So that your Bacillus stearothermophilus, is a heat-loving microbe. Kaya siya nilalagay na pang test sa autoclave ko minsan. Kasi ang autoclave mo, high temperature yan para nga mapatay mo yung microbes. And then, ipapakita ng bacillus mo na na-reach nung autoclave yung ganong temperature kasi mag-grow siya at that temperature. Okay? So, mag-change ang color ng vial na pinaglagyan yan, yung mga iba-ibang itsura ng biological indicator sa autoclave. So, you'll learn more about that when we get into disinfection and sterilization. It's just that because your Bacillus stearothermophilus is the given example here, it is a heat-loving organism. Kaya yan ang ginagamit, high temperature ang gusto niya na environment. Then you have your hyperthermophilic, above the temperature of boiling water. So that who can explain that? Giselle, para tanggalin ko yung ex ko dito, Giselle. Nag-freeze pala ako kanina, akala ko hindi kayo wala. Giselle, where's Giselle? When we say above the temperature of boiling water.
Ma'am, they thrive above 100 degrees Celsius po.
And what temperature would that be?
Above 100 degrees Celsius po.
Above 100 degrees Celsius. Okay. So, how about your extremophiles, Giselle?
Sa niya, ma'am, they are able to survive in extreme temperatures naman po.
So, it's either very high or?
Yes, or very low po.
Or
even
low.
absence po of oxygen.
Okay. Okay. Yes. So, that is as to temperature. Now, this is just a graph of how well showing you the different temperatures and the growth rate of different organisms, showing you whether they are cyclophile, cyclotroph, mesophile, thermophile, or hyperthermophile. Okay, so ito yung sinasabi ko kanina because you have a minimum, you have a maximum, then you have your optimal. So that when we say minimum, di ba may range ka pang 0 to 10, grows well at 0 to 20. So that your minimum would be, parang dun sa inyong psychrophile. They grow well at 0 degrees centigrade to 20 degrees centigrade. What would be your minimum growth temperature?
Hello, ma'am.
Ito, yeah. Sino to? Hello.
Jonavi, ma'am.
Jonavi, para matanggal yung ano. What would be your
minimum range po natin kapag cyclophile is 0 degrees Celsius. Tapos, yung maximum would be the 20 degrees Celsius.
Okay. So that when we say optimal, teka, nalulubat ako. Optimal, Junavi, tuloy-tuloy mo na siya. Optimal.
Optimal range po at 20 degrees Celsius, ma'am.
Optimal temperature at which species grow best. Okay. Yung highest, so it could be yung ano mo usually. Kaya sabi ko kanina, tignan nyo yung most organisms are mesophilic. So that in 30 degrees centigrade, it is the optimal temperature for many free living forms, and the body temperature of the host is optimal. Okay, yun yung pinakamaganda nila. Now, aside from this maximum growth temperature, optimum, and your maximum, meron din tayong tinatawag na thermal death time, and you have your thermal death point. Okay, so that your thermal death time is the lowest or minimum time required to kill organisms under constant temperature. Like for example, kung nung unang panahon, wala pa tayong bakit nag-freeze ang aking nag-iba yung, ano ba yan? Wait lang. Sorry. Mag-off yata yung laptop ko. Wait lang, para nag-freeze yung aking laptop. May ikot siya, hindi ko alam kung mag-off siya or... Ano gagawin ko dito? Okay, ito na. Okay. Aron na. Ayaw pa din. Sabi natin kanina, thermal death time, lowest or minimum time. Take note ha, it's the minimum time required to kill organisms under constant pressure, temperature. Like noong unang panahon, nung wala pa yung mga mineral water or aside from mineral water, yung mga filtered ba yan? Or whatever. Di ba binoboil ang water for 15 minutes, boiling yan, to get rid of the coliform organism. So the thermal death time of that E. coli, which may be present in water, is, what is the minimum time? It is 15 minutes. Whereas, when we talk of thermal death point, that would be the lowest temperature. So we're talking of temperature here. So ang temperature natin would be, saan natin start ma-eliminate si coliform? Sa boiling point niya is, mag-start lang na mamamatay yun pag nagbo-boil na siya. So that would be like 100 degrees centigrade. Okay. So we proceed now into this again, that you always take note that diagnostic laboratory usually incubate cultures for bacterial growth at 37 degrees centigrade. Although Pseudomonas aeruginosa and Campylobacter, remember I mentioned Pseudomonas aeruginosa earlier with E. coli, having very simple requirements, and then they can be easily grown in the the laboratory, so that you see here that they can grow at 35 degrees and 42 degrees centigrade. So, we proceed now to your next physical requirement of our organisms is the pH. Ano yung pH? Ano ibig sabihin ng pH natin? pH.
Ma'am.
Yeah, sino to?
Ma'am, Israel Gatcho po.
Hindi ka pa nag-recite, Israel? Teka muna, tingnan.
Not yet.
Sige, go.
Ma'am, pH is the measure, ma'am, of the hydrogen ion in a solution, ma'am.
Okay.
Or it's... Ay, lang po, ma'am.
Okay, yes, it is the hydrogen ion concentration. And of course, in this pH requirement, meron na naman tayong classification ng ating mga bacteria. Depende na naman sa pH na gusto nila. Wherein you have the following. We will discuss it with ituloy ko na, konti na lang naman, hanggang na lang yun, ilang slides na lang. So that, yung pH niya, alam niyo naman ang pH is below 7, that would be your, na pag below 7? Your?
Acidic, ma'am.
Acidic. So that when they're acidic, that would be your acidophiles. Above 7 will be your
Alkali
Basic,
file.
ma'am.
alkaliphile. So that pag siya ay 7, siya ay neutrophils.
Neutralophile.
Okay. So, doon lang naman yung most para lahat karamihan ng media natin, culture media, they're at a neutral pH. Kasi there are those bacteria, paano nyo ma-maintain ang neutral pH? Paano nyo ma-maintain ang neutral pH? Para hindi siya, meron siya maging stable ang pH niya. Kahit lagyan mo yan ng something acidic, meron siyang mechanism of trying to bring back the pH to 7 just to keep it at a neutral range. Anong mga ginagamit nyo doon?
Ano, ma'am?
Yes, Tam. Ang init, Tam.
Buffer solutions, ma'am.
Buffer solutions. Okay, very good. So, yung mga yan, malalaman nyo din later on, pag nag-AST tayo, antimicrobial susceptibility testing, that one of the characteristic of your Mueller-Hinton agar, the one that we're using for antimicrobial susceptibility test, is a constant buffer. Okay? A constant buffer is yung constant pH niya. So aside from now, we discuss temperature, and then we discuss about the pH or the hydrogen ion concentration. Now we go to osmotic pressure. So sabi natin, what is osmotic pressure? Ano naman yung osmotic pressure? This is talking about what? Yung pa, hindi pa naka-recite, hindi ko pa naman dala yung ballpen ko, lumabas ako na nandito ako. 3G lang sa kwarto, hindi siya makapag-LTE. Ano naman pag anong osmotic pressure? Mag-memorize na lang ako. Sino? O si Godwin, kasi si Umayam yun kanina, di ba? O si Godwin Tabalbag. Ano naman ang osmotic pressure?
Ma'am, yun yung pressure na kailangan, ay minimum pressure na kailangan para ma-prevent yung inward flow ng solvent papunta sa membrane.
Ano yung process when we're talking of osmotic pressure?
Osmosis, ma'am.
Osmosis. So that ito na yung kinokontrol ng cell membrane. Which microorganisms have a freely permeable membrane? Are they the gram-positive or your gram-negative? Going back to your bacterial structure. Sino ang mas permeable?
Positive.
Yung gram-positive. Okay. So that if they are, what do you call the organisms now requiring high osmotic pressure? They would be called your, when we're talking of osmotic pressure, and there are organisms which require high osmotic pressure just to make sure nothing goes out or nothing comes into the bacterial cell. That would be called your
Osmophiles, ma'am. Osmophilic.
osmophiles, or there are their osmophilic. Okay, so yun ang ating physical requirements, just so that we get into it na. Physical requirements, you have your temperature, so you have there commonly 3 lang, because you have your extremes, you have your cyclophilic, you have your mesophilic, and then you have your thermophilic. Of course, wala yung mga extremes doon. Then you have under your hydrogen ion pH, you have your acidophiles, you have your neutrophiles, or your alkaliphiles. Then for osmotic pressure, you have your osmophilic. Now we get into the bacterial growth curve. Okay. Anyone familiar with the bacterial growth curve? What is this bacterial growth curve? Junavi, dalawa din account mo? Judrit. Judrit yata nag-perfect ng drill. Judrit, alam mo na yun sa bacterial growth curve? Yes, Judrit. Hindi kita marinig. Judrit? O, Kathleen. O, teka, nag-on si Judrit. Judrit. What is the bacterial growth curve? Wala kang audio, di kita marinig, Judrick. Yun yung kanina ulit, meron. O, yan. Nawala ulit. Judrick? Wala. O, wala kang audio. Medyo anuhin mo. Ano yung bacterial growth curve? Ano ang pinapakita doon?
Ma'am, ma'am, the, ano po, the ability, first, ma'am, I think po,
Ah?
the capacity of the, the bacteria.
Dalin. Ayusin mo yung mic mo, Judrick. Nawala ka. Okay, what is the bacterial growth curve? This one, wala na ang ko. Ito, yung characteristic growth curve is obtained when plinat mo yung sa logarithm of the number of cells against the time of growth. So remember that we're talking of the number of cells when we're talking about the growth of our prokaryotic microorganism. So that meron tayong 4 phases sa growth curve. But this can be achieved only if the microorganism is in a balanced environment. So that when we say balance ang environment niya, provided lahat ng kailangan niya. Lahat ng kanyang chemical requirements, lahat ng kanyang physical requirements. So, ano yung 4 phases na yun? So that we have the log phase or your LAG phase, and then you have your exponential or your logarithmic phase, and then you have your stationary phase and the death phase. Dun sa log phase, your LAG, or this is also called your phase of rejuvenescence or yung phase of physiologic youth. Bakit tinatawag ito na phase of physiologic youth or yung rejuvenescence? Kasi dito sa period na ito, siyempre dito pa lang, nagtatry na mag-adapt yung organism sa kanyang new environment. So during this time, pwede itong makarakterize ng walang multiplication o meron ng konti. Meron ng konti. But more so is, siyempre nag-a-adjust pa lang siya dun sa environment niya. Hindi pa siya talaga nagmo-multiply. Okay, again, going back, bakit multiplication? Because we said that growth of your prokaryotic is to increasing their number. Okay, so that yung mga cells sa phase na ito, they're already metabolically active. So meron ng active synthesis ng mga enzymes and other yung mga essential constituents dun sa kanilang environment. Okay? So that after a while, naka-adjust na siya, bigla ka magkakaroon ng exponential or the logarithmic phase. Dito yung ano na, siyempre naka-adjust na sila sa temperature nila, nandito na yung mga ano nandun na sila sa stage of cell division, tsaka meron na yung logarithmic, kaya nga tinawag na logarithmic phase. Yan yung meron na siyang mabilisan, dumadami yung number nila. So, merong steep na curve yun. Pag-check nyo sa notes ninyo yung logarithmic phase. Dito sa phase na ito, yung generation time niya ay constant. Okay, so di ba sabi natin pag they reproduce by mitosis that one cell will become two, and then yung generation time niya is constant. Then after that logarithmic phase, makikita mo biglang may plateau. So, imagine niyo na lang yun. Kung meron kayo, makita niyo sa notes nyo naman, merong plateau. Yan na yung, dito sa period na ito, yung self-reproduction now equals the rate of cell death. Kasi after ng stationary phase, meron naman yung downward phase natin, yung pababa siya, which is the death phase. But during the stationary phase, bakit nagkaroon ng stationary phase? Bakit hindi na sila nag-multiply o hindi na sila nag-reproduce? Unang-una, syempre, nagamit na nila lahat yung nutrients na nandun. Syempre, dumami na sila, nag-start sila, kunyari, ilan lang sila. Nag-adjust sila sa temperature, naka-adjust na sila, nag-multiply na sila. Dumami na sila, dumami nang gumagamit ng nutrients. Naubusan ka na ngayon ng nutrients. At dumami na sila, pag gumagamit sila, i-ano natin sa situation natin, na pag may input tayo, kakaroon ka ng output. So that yung output ng mga bacteria na yun, nag-accumulate din ng waste products, which contributes to a toxic environment. And those waste products, magkakaroon na rin ngayon ng change sa pH ng environment. So it could either become now an acidophilic environment or alkalophilic. Sorry. Magiging alkaline na yung environment nila, which may not be favorable to the organism. That will now lead to the last phase of your growth curve, which is the phase of decline or yun na yung death phase. Ito na yung period na merong complete cessation of multiplication. Kasi meron na ngayon meron ng increase sa death rate ng organism. So, pababa na siya. So, yung una niya is your lag phase. Flat pa lang yung curve mo. Naka-adjust na siya. Nagkaroon ka ng exponential. May steep increase na siya. And then, magpla-plateau siya. After the plateau, decline. Okay? So, that is your growth curve. Now, there are also other measurements of bacterial growth wherein we can try to do ano na yung mga nandun sa ating PowerPoint. Hindi ko matandaan, hindi ko na ma-open. Meron kayong 4-plate technique or yung spread technique. Yung 4-plate, familiar na kayo dun sa Petri dish, di ba? Yung 4-plate, yung pinaglalagyan natin ng culture media where we try to grow the microorganism. Pag 4-plate, malambot pa yung media natin before it completely solidifies. You put now the culture broth into the plate and then pag medyo malamig na yung media pero hindi pa siya magso-solidify, you pour the media onto the plate with the microorganism already that has been serially diluted. May serial dilution din. Kasi pag concentrated yan at pinor natin dun, hindi rin natin makikita yung individual growth ng microorganisms. Whereby, hindi natin sila makikita, marami sila na ganun. Magkakaroon tayo, hindi mo sila mabibilang per colony. And when we're using this one, ang unit of measurements when we're counting microorganisms is we're counting them as colony forming units or number of bacteria per ml. Kasi may computation din dyan na pag gumamit tayo ng serial dilution, so like for example, you do a 10-fold dilution, nag-dilute ka ng, you had your original bacterial culture, wherein you will get 1 ml of that, you transfer it into 9 ml na bacterial broth media. Okay? So, your dilution there will be 1:10. So, from the 1:10, kumuha ka ulit ng 1 ml, nilipat mo ulit sa 9 ml. So, the volume will always be the same throughout. Then, from the 1:10, since it's a 10-fold dilution, the next dilution will be 1:100. And then, you dilute that further. That will be 1:1000, then 1:10,000, and so on and so forth. So that when you try to compute for the number of cells that may be present, then you count the colonies formed doon sa plate ninyo times the number of yung sa dilution niya, kung 10,000 ba yan o 100,000. So nakabilang ka ng 10 na colonies and your dilution was 1:10,000, that will be 10 times 10,000, that will be the number of bacteria per ml of your bacterial culture. Okay? Now, meron din dyan yung parang ah, cell counter ninyo sa microbiology, yung Neubauer counter, yung pinagbibilang ng red cells and white blood cells. Meron din ganun na ginagamit sa microbiology. Now, another measurement would be the measurement of turbidity. Kasi ang indicator na nagkaroon ng bacterial growth into your broth media is that your broth media will turn turbid. So that meron yung pag masyado siyang turbid, ang density ng microorganism, marami sila. And the lighter it becomes, kumukonti yung number ng bacteria. So how do you measure for this one? Pag turbidity ang ginagamit, mini-measure yan sa spectrophotometer. And application ng turbidity is when we will be doing our AST, when we will be doing our standard, you have to compare bacterial growth over this standard para alam natin na nare-reach natin yung 1 times 10 to the 8 colony forming units when we're doing our antimicrobial. Yeah.
Ma'am, hala na po kaming maintindihan kasi walang screen.
Wala nang screen. Hindi nyo na siya nagigets. Okay. Di ano ko na lang. Anyway, last na lang naman yung ito na lang measurement of bacterial growth. Kasi yung bacterial cultivation sa lab natin kukunin. Okay? So, i-present ko na lang ulit siya sa ating, we'll start from, represent ko pa ba ulit yung bacterial growth next meeting sa Thursday? Andyan yung curve sa ano nyo, sa notes ninyo. Hindi nyo naman yata binabasa ang notes ninyo sa module ninyo. Kasi yung PowerPoint galing lang dyan sa module nyo na-summarize lang siya. Para lang ma-lecture siya. So anyway, mahirap nga siya i-imagine, I know. And we'll just proceed with this on Thursday. Thursday, ano, Tam?
Ma'am, Wednesday po.
Wednesday ba?
Wednesday
Bukas.
po.
Sige. Itapusin lang din natin hanggang sa measurement of bacterial